Muy buenos días a todos. Bueno, pues nos da mucho gusto estar con ustedes ahora y es la nueva forma de los seminarios de Frontiers in Genomics que antes teníamos los martes en la tarde. Ya este semestre por lo menos los vamos a tener los martes en la mañana. Como ya indiqué en el mensaje de TAB que les hice llegar a la mayoría de ustedes, es que los seminarios tendrán horarios un poquito variables en los martes en la mañana y serán entre las 10 y las 12 del día. Y bueno, pues el día de hoy tenemos con muchísimo gusto la participación del doctor Daniel Geschwind y él es profesor de neurología y psiquiatría y genética humana de la Facultad de Medicina de la UCLA. Y también es el codirector del Centro para Genética del Neurocomportamiento, también en la misma universidad. La formación del doctor Geschwind es muy interesante porque está realmente de una manera muy completa. Se ha dedicado a estudiar la neurobiología, él es médico y es internista, pero por otro lado tiene una especialidad en neurología y por sus estudios de licenciatura también tiene una licenciatura en química y psicología. Entonces, todo esto hace que tiene una formación extraordinariamente completa para estudiar la neurología y la genética de muchos padecimientos humanos. Su laboratorio combina muchas técnicas de genética, de genómica y de bioinformática para justamente abordar, y modelos obviamente in vivo e in vitro, para abordar todas estas enfermedades del sistema nervioso central, del sistema nervioso, para entender cómo es que funciona, cuáles son los genes que son responsables de los padecimientos y para desarrollar nuevas formas de terapias que sean útiles y que se puedan aplicar en un futuro cercano. El doctor Geschwind es un investigador con realmente un impacto muy grande en su campo y participa también, ya no como investigador, sino que realmente como un consultor en diferentes instituciones, tanto públicas como privadas. Es un miembro de la Academia de Medicina y también pues es además una persona extraordinariamente amable y muy abierto a la discusión, como pudimos ver ayer con los estudiantes de la licenciatura en Ciencias Genómicas. Daniel, I would like to thank you very much for coming or being with us, at least today. And we really appreciate the effort that you are doing to participate in this program.
Thank you. Muchas gracias. And it's really my pleasure to be here virtually. I wish I could be there in person. And maybe someday we'll be in Los Angeles or in Cuernavaca. But
maybe.
Yeah,
We hope so.
yeah, hopefully we'll all be out of our houses in a few months. Although I have to say, UCLA is not really planning for on-campus activities for fully open until March 19th, which would be 1 year. So anyway, but on to hopefully things that we'll be able to get back to. The title of my talk is Systems Biology Identifies New Mechanistic Targets for Neuroscience Therapeutics. And the idea here is that we use these integrative genomics approaches to identify mechanisms. And by understanding mechanism, we can develop therapeutics. But as I'm going to show you, that we can also directly just target the genomic networks. And that seems to work even if we don't know what the exact mechanism is in some cases. So, you know, essentially, so let me really get started. Here are my consulting arrangements. That's kind of a disclosure, but the stuff I'm talking about today really isn't related to any of this other than Takeda, which funded which gave us direct support for research, and I'll describe some of that. So I'm going to organize my talk around 3 problems. The first is, can we use transcriptomics to define a molecular pathology of autism and neuropsychiatric disorders? The reason why that's such an important question is that, as opposed to neurologic disorders like Alzheimer's or Parkinson's, there's no imaging, gross pathological, or even microscopic pathological alterations visible in people with autism or schizophrenia or bipolar or major depression. But yet we reason that perhaps looking even submicroscopically at RNA, we'd be able to define a molecular pathology. Problem 2 and 3 are problems that we've been working on a very long time, kind of in the background in a way, and I think they're really coming to fruition. One question is, why do peripheral nerves regenerate and central nerves do not? And problem 3 is, what are the drivers of neurodegenerative dementias? The reason why so we've asked all of these questions, what really ties them together is the approach that we're using, and hopefully you'll see how they inform each other. The second 2 problems I show because there we are able to actually move from networks and systems biology to finding drugs that reverse the phenotypes of interest. So in problem 2, we can show that we can find drugs that increase central nervous system regeneration. And in problem 3, we can find things that block neurodegeneration that are predicted to by the systems biology approach. Problem 1, we're just at the point of defining the systems biology, the networks, but we don't know what phenotypes we have to reverse. It's not cell death. Is it more synapses, fewer synapses, more excitation? We don't know that yet. And hopefully over the next 5 years, we'll get to that in basic neurobiologic studies. And then we'll be able to do for problem 1 what we've done for problem 2 and 3. So genetics is a simple paradigm, and it starts with causality. And the major point I want to make here is that there's a huge genetic contribution that's been recognized for a while for the psychiatric disorders, starting with anxiety at the top, and even schizophrenia and autism towards the bottom there. The point is, these are very frequent diseases, so there's huge burden on society. They're very heritable. And so the thought was, even 30 years ago, genetic studies will yield fruit. And finally, in the last decade, that has really come true as the studies get large enough. And the idea is we find loci, we find genes, and determine how they cause disease. And today, I'm going to really talk mostly about understanding mechanisms, the third. But my laboratory works across all 3 of these because it's a very clear and logical way to approach disease. One of the points here that I want to make is that, we think of these psychiatric, 2 things. One is psychiatric disorders aren't defined by a blood test or an image or even pathologically. So they're defined by behavior and cognition. And therefore, there's some uncertainty around the diagnoses. And so it was unclear for a long time whether they were refined enough diagnoses to actually find genes. That's turned out to be the case, that they are refined sufficiently so that when you get large enough samples, you find a lot of loci across the genome. For schizophrenia, they're close to 200, autism over 100, et cetera. But one of the most interesting things that we learned from looking at the SNP-based heritability, that is the common genetic variation that contributes to these disorders, which is only one component of the disorders, is that there's substantial genetic correlation across disorders. Now, maybe that doesn't surprise you for schizophrenia and bipolar, which are often misdiagnosed, especially early on. But even for autism and schizophrenia, there's a 0.2 genetic correlation. It means that most of the variance is separate genetic things, but there's still substantial overlap. And so we've been quite interested, and we have to keep that in mind as we're studying these disorders. And so what we found hundreds of genes. There's genetic heterogeneity. For autism, there's no single gene that accounts for more than 1% of autism. Same thing in schizophrenia. It's extremely, and this kind of notion of polygenicity, that of multiple variants mostly of small effect sizes, but some of large effect sizes. But genes don't act in isolation. They're components of biological signaling pathways, and in the brain, complex neural networks. Can we identify patterns within these large dimensional data sets? So as we're measuring genomic things that are related to key aspects of biological function or disease pathophysiology, and can we use these patterns that we measure to inform treatment development? So the notion is, rather than just collecting one piece of information and looking at it, we're going to be looking at things more systematically and trying to combine different levels of analysis. So one of the things that really drives our thinking is, I mentioned genes don't act in isolation. Well, then the question is, what is the structure of the networks or pathways that we should expect to find as we're studying these disorders? So work by a number of people, and I'm showing some slides from Laszlo Barabasi, who's a physicist, really push this notion of scale-free networks. And to a first approximation, most living systems are self-organizing systems that obey this notion of scale-free topology. And what that means is that a small number of nodes have a large number of connections. Most nodes have very few connections, and the few highly connected nodes are essential for the integrity of the network. So you can find these kind of highly connected nodes or hubs, and they kind of turn out to be important drivers. Now, initially this was shown for protein-protein interactions in yeast, and working with Steve Horvath at UCLA, who is a statistical geneticist who developed the method WGCNA that we use, we showed that gene expression, even though also obeyed scale-free topology. And so our notion is, we're thinking about networks, we're thinking that genes don't act alone, and we're trying to connect these different levels. So we have to go from genetic variation, which is causal, all the way up to the brain. And what I'm going to be talking about mostly in this talk is measurement of mRNA or even microRNA, but RNA species, not because it's the be-all and end-all, but because it's a tractable phenotype. I can measure it inexpensively in high throughput with almost complete dynamic range down to 1 copy per cell. So if I could measure protein in that same way, I would be doing that as well, no question. But the transcriptome gives us a view that we can't get from the other systems because we can't measure it in the same way. But the notion is that at all these different levels, I showed you the internet and social systems are scale-free. The notion is these circuits and synapses are going to be scale-free, as is the mRNA. And the issue is, how do we kind of make these networks? And this isn't a talk about how, comparative network analysis, but this is just, so I'm not going to discuss that in detail, although I'm happy to answer any questions. But because genes don't act independently, one can use their kind of mutual dependence or correlation. There are a lot of different measures you can use. Here I'm showing correlation. And this is just a cartoon of expression levels on the y-axis, samples on the x-axis. And we can see that the genes fall into groups or modules or clusters based on their co-expression, based on their covariance. From there, we can identify these clusters. We can show that they're reproducible and robust. And within the cluster, we can identify hub genes, that is, things with the very, very high number of connections that are at the center of the module. And what's shown here is a cartoon showing what happens if we took 100 samples of cerebral cortex, actually, and we did this off the internet from other laboratories, and ran and tried to measure the correlation substructure, creating modules, and then analyzing those modules. And those modules correspond to basic elements of biology. Here, if we take whole cortex, the major source of variance in that tissue is cell type. And so we pull out modules that correspond to different kinds of cells and different components of cells if we do a PCA analysis. That's just shown here. And so we can do an in silico dissection. In other words, if we have whole tissue, we can actually make inferences about the cell types that are in that as well, even without dissecting them out. So we use this methodology, starting with the correlation, making modules, understanding the network structure, and then annotating the modules to figure out what they are. to get to some biological insight. So the questions in this part of the talk: can we use transcriptional networks to characterize the molecular pathology of a neuropsychiatric disorder, autism? And then, can we use this framework as a quantitative framework for cross-disorder comparisons? The answer to both of these is yes. We've published on this, and so I'm going to summarize it quite quickly. The first study was driven by Irina Buonago, who applied an unsupervised network analysis based on let's just say, in the 19 cases and controls, cerebral cortex, temporal and frontal lobe from autism and controls that were relatively matched for age and sex. And she ran network analysis and then compared the modules. And we identified the same modules in autism and controls, but their trajectories were different. So we go from 10,000 genes measured at that point on a microarray to 20 modules. So you can see another advantage is that it's grouping genes into biological pathways and cell types, but it also reduces the dimensionality of the data. And what's shown here is a multidimensional scaling of the modules, which are colored and numbered. We also put in this covariates like region, sex, postmortem interval, age, and then whether or not you have an autism diagnosis. And you can see that there turn out to be two modules, M16 and M12, that are changing in those with autism relative to controls that are related to the autism diagnosis. Those are two modules. And it turns out what I'm showing here are the autistic individuals on the left and the controls on the right in a heat map where green is down and red is up. Each on the y-axis is a different gene. So what it's showing, and then I've shown the first principal component for ASD-M12. And you can see that in about 2/3 of samples, it's down in the autism and not in the controls. And similarly, ASD-M16 is up in the autistic individuals. So now we go and annotate these modules, and it turns out that ASD-M16 is involved in synaptic transmission, vesicle recycling, energetics, things related to neuronal function and synapses, whereas ASD-M16 is annotated as a neural immune module in gene ontology, and turns out to represent microglia and astrocytes. So that was in 2011, and that was the first time anybody had really shown that there was a consistent molecular pathology across a very genetically heterogeneous disorder. It's really surprising to us. Small sample size. So now we increase the sample size almost by a factor of 5. We now have 113 cases and controls. We use much more complete profiling using RNA sequencing and microRNA sequencing. Here, I'm just talking about the mRNA. And because we have more samples, we get more resolution. We're able to divide the neuronal modules into 3 that are downregulated, shown here in A, and then 2 that are upregulated, M19 and M9. Here, it's separated the microglia into one component and the astrocytes into another. And that's shown in B, where we basically ask, are any of the modules enriched in markers for cell types? So M19 being a microglia, M9 being astrocyte, et cetera, and these 3 being primarily neuronal. There's a lot in this 2016 paper, and I think if you're interested, you should take a look at it, because there's also a lot with regard to quality control of transcriptomic data that I think is of general use to anybody using these methods. Emily Wu, another postdoc in the lab, led a study of the microRNA and showed a very similar pattern. And this is the red being autism and the black being controls, that it wasn't complete separation of them, but one could see that there was a pattern that was present, again, in about 2/3 of cases and controls. And some of these microRNAs we could show were actually driving some of the neuronal modules that we saw there. Further, we collaborated with Shyam Prabhakar to show that we wanted to understand, okay, what is the nature of the transcriptomic changes? Is it being driven by changes at the chromatin level? So the first step was to look at active enhancers using H3K27 acetylation ChIP-seq, which we did in frontal cortex and temporal lobe. And we saw a big overlap between frontal cortex and temporal lobe. And as in our previous studies, we saw a pretty minor signal from cerebellum. It's mostly going on in the cortex. And these peaks overlap strongly with gene expression changes that were occurring in the brain. So we could see that, there was a correlation. One of the key points of this that I want to make is that these H3K27 acetylation, microRNA, and other things we've looked at, RNA degrades and there are confounders, and we tried to control for them as best as we could. And we show, I think fairly convincingly, that we do. But here are other molecular methods that don't have the same kind of confounders, the same kind of degradation, and they're really showing the same thing. So there's a kind of convergent biology here that we've identified. And we just had a paper that came out in Nature Communications that integrates all of these data and shows that there's kind of an integrated signal. So now where are we? And again, this is, very, very high level. We're not at the level of mechanism yet. But, for almost a half a century, we've known that cancer is a disorder of proliferation and invasion, that stroke is a disorder of acute brain hypooxygenation. All of these things have many, many genetic and environmental contributions. In this context, what is autism? And we're just trying to get to that very primitive level. And so what we know is that there's decreased neuronal gene expression, increased microglia and astrocyte gene expression. And we showed in some work that I'm not going to show you, but that is in these published papers, that actually there's alternative splicing defect driving some of the altered neuronal gene expression. And we want to now understand what's causal, right? Because, we're looking at brains from people who had autism who died from age 4 to 51 in the first study, and even more broadly in the second study. And so it could be that the autism, we're not saying that these patterns are the cause of the disorder. This is just a pathology. It's the state of the brain. And now we have to understand how it gets there. And the question is, where does the causal genetic drivers sit? Are they driving microglia and astrocytes or the neuronal gene expression? And it's crystal clear that the genetic variation that's been identified to date is driving the neuronal dysfunction, and that the kind of microglia astrocyte, what's called neuroinflammation, is really part of a synaptic dyshomeostasis that's ongoing, and that it might originally be adaptive, but chronically, we presume or hypothesize, probably maladaptive. How do these changes in autism relate to other psychiatric disorders? Are these, is it specific to autism? And so again, the notion that Mike Gandel, when he was a postdoc in the lab, he now has his own lab. We have these different clinical syndromes. You have different etiologies, highly, genetic, but and then you have brain circuit dysfunction that the patient has, and an altered brain transcriptome, which is this, again, intermediate phenotype. And again, going back to the coheritability, this is from a seminal paper from the Psychiatric Genetics Consortium, again, showing that even things like, schizophrenia and autism have a kind of, substantial coheritabilities. So we're asking, is this coheritability map onto the molecular pathology of these disorders? So Mike spent almost a little more than a year, almost like 14 months, finding data sets, normalizing them in multiple different ways to ensure the QC and balancing was essential here. So in the end, we had 807 samples from 6 different diseases and 15 studies, and controls. We just looked at cerebral cortex. He performed analysis of differential gene expression and ran network analysis. So when we just look at the global transcriptome and ask how are the changes correlated across disorders, we could see that schizophrenia and bipolar were the most strongly correlated. They were modestly correlated with depression. It was significant, however, and even more correlated, schizophrenia being the most correlated with autism. There was a small correlation of autism and inflammatory bowel disease that didn't reach significance, but I bring it up because it reflects the fact that the same kind of cytokines that are expressed in other peripheral inflammatory conditions are also used in the nervous system, but they have different functions. And that's where this overlap is. We had alcoholism didn't relate to any of these disorders. And that also, aside from the fact that we were very, very careful about taking out batch effects, confounders, clinical issues, postmortem interval, the fact that, alcoholics have seizures, they have the same postmortem issues, and yet there's no correlation between the alcoholism and the others. What was really striking to us, though, however, really remarkable, is that when we actually asked in a pairwise sense, using both microarrays and we created new RNA-seq data from a small cohort of autism, schizophrenia, and bipolar disease as part of PsycEncode with Kevin White and colleagues, we were able to see that both at the RNA-seq and microarray level, that the pairwise transcriptome correlation across diseases was strongly correlated with the SNP-based correlation of disorders. And the rho was extraordinarily high. The Spearman, so almost 50% of the variance in this relationship can be explained by genetics, which I thought was really kind of remarkable. This doesn't, again, mean that genetic variants are acting as expression quantitative trait loci. That is, the SNPs are changing the gene expression. It just means that there's an underlying biology that occurs, and that's partially shared, and it's related to the genetic overlap. So this really showed how one can begin to use, and one of the issues in these disorders is, how do you compare a 4-year-old or 5-year-old or 9-year-old autistic to a 50-year-old schizophrenic, right? Different cognitive batteries, different brain imaging. The transcriptome kind of provides a nice, very, very quantitative and molecularly tractable phenotype for this. When we do network analysis, when Mike Gandel ran network analysis, he identified, this is just showing, again, it's a multidimensional scaling of plot, similar to the one I showed you before in autism. Now we show the modules cluster by their cell types. Again, we have 4 neuronal modules, 3 kind of glial modules, and they're anti-correlated with each other as shown by these lines, red being anti-correlated, green being positively correlated. And I'm just gonna, again, show you just 3 of the disorders here. Let's pay attention to autism and schizophrenia to start with. Autism, the 4 neuronal modules are downregulated, shown here, and then there are 2 upregulated that turn out to be the CD11 and CD4, the green, yellow, and the yellow. The yellow turns out to be astrocyte, and the green, yellow, microglia. We look at schizophrenia, we see a similar pattern, although much more attenuated. We see astrocytes upregulated, but we do not see the microglia component. We don't see that in bipolar either. And alcohol abuse disorder looks entirely different than all of these. When we look for the genetic enrichment, we find genetic enrichment is in several of the neuronal modules and not in the glia, again, supporting the notion that primary genetics is is driving neuronal dysfunction. Now, I have to say for the schizophrenia, we took out the MHC, which is such a large signal. And so there's no question that there's a neural immune component of schizophrenia. But we had just taken out the MHC for this analysis because it so dwarfs everything else. So, these are, those are studies where we, took data from the web and correlated them and got them, and it's all microarray. And so, at the kind of in parallel with this, as this work was, it took us almost a year to get that other paper published, more than a year, about 18 months. We were working as part of PsycEncode. And PsycEncode is a huge scale, multi-lab collaboration, multi-institutional. We're trying to profile brains of folks with mental illness, as well as define the regulatory networks that are dysfunctional. And so here we had 51 autism, over 500 schizophrenia, bipolar, and nearly 1,000 controls. And this gave, and we have RNA sequencing across all of these. We can do the analysis at the looking at splicing, looking at transcripts, looking at isoforms. We did a network level integration. There's this paper, this one transcriptome paper that's part of a package of about 6 or 7 papers that were in Science. Really it has kind of set the framework for another decade of work for us, as you'll see on one of the next slides. But it largely validates, but substantially refines what we had seen before. In fact, we could see very specific neural immune trajectories, again, where autism has this microglia upregulation while the others don't. But then it also pulled apart interferon response and NF-kappa B responses as being quite distinct in these disorders. In fact, the interferon response module had something like, I don't know, 59 of 62 known interferon response genes, and the NF-kappa B had 4 out of the 5 NF-kappa B members. So these really do represent those signaling pathways. Just to give you an idea, here are the isoform and gene modules identified. So now we go from kind of a few dozen modules associated with disease to 96 modules, 60 of them are associated with disease. And so we show the disease effect here. There's cell type enrichment, in other words, what kind of cells they might represent, as well as enrichment for GWAS, genome-wide association. And so we can see that, this was really an exquisite level of analysis, and it was extremely difficult to compile, and kind of it took us more than a year to make sense of it. But basically, you can organize the modules by their kind of cell types and biological processes. And this, again, is just a subset of the major things that are associated with disease. And so we can see blue is downregulated, red is upregulated. The circle around things that's red shows a GWAS enrichment, either nominally or FDR corrected. And essentially what you can see is some things like NF-kappa B have a gradient across these disorders. Others, like the interferon response, are just literally really observed in one and not the others. Other things are observed in, in schizophrenia and bipolar here and not in autism, et cetera. So we've really been able to refine the signal. We can refine across different kinds of excitatory and inhibitory neuron signals. And again, we have a really strong downregulation of interneuron genes in autism that's not seen as much in the other disorders, et cetera. So in conclusion, integrative genomic and gene network approaches provide an organizing principle for understanding molecular phenotypes in the brain and define points of potential biological convergence in disease. Now, what I just showed you, there are numerous of those modules that we're now trying to work on and understand what's biologically driving them, especially those that are enriched in genetic signal. RNA and epigenetic profiling define robust molecular pathologies of major psychiatric disorders, and these define specific and overlapping pathways that are a starting point for mechanistic understanding and therapeutic development. And of course, as I mentioned to you in the beginning, knowing what cell biological circuit phenotypes that we're trying to modulate and in what direction is essential to drive further therapeutic development. So now I'm going to talk about the next 2 points. So here, we kind of stopped with a transcriptional network. And what I'm going to show you in problem 2 and problem 3 is that we identified the transcriptional network and then used that to identify drugs that are predicted to reverse the network or to enhance it. And then we show that it actually has that phenotype effect. I'm going to go through pretty quickly. So step 1, you characterize the network modules associated with a phenotype. You identify drugs and genes predicted to change expression of the module genes. And you can do this because there are databases out there like CMAP and CLUE at the Broad that allow you to do this, that have been built. We test if predicted perturbation drives the changes, and then we test if the predicted drug has the desired phenotype effect. So here, the first study that was published in 2016 in Neuron was led by Giovanni Coppola and Vijay Chandran, who both, Giovanni had been running his own lab for quite a while. Now he runs a part of a group at the Regeneration Genetics Center, and Vijay Chandran has a lab at University of Florida. Here's the problem. I'm showing you a dorsal root ganglion cell here. This is this cell is a sensory neuron that is in the sciatic nerve, and its peripheral part goes in the sciatic nerve. When you cut this, as long as you keep the sheath around the nerve or you crush it, it will regrow. Same cell, if you cut it in the central nervous system, doesn't regrow. Same thing with the optic nerve. So central nervous system doesn't regrow, peripheral can if given the right tube to grow through. So we took data from multiple collaborators, Mark Dzinski, Mike Fanzelberg, Clifford Wolf. This was an international collaboration where they had done lesion studies of sciatic nerve and some of the spinal cord. And we could compare them over time and ask what is different about the transcriptional response. And so we created a regenerated associated gene network. And what's shown here, this is from the Dzinski data set. This is from the Fanzelberg. This is a after we've done the network analysis, we identify modules, and this is showing how the modules cluster in each separate data set done in different labs at different times. And this red is the overlap of the similarity between the clusters. In other words, there's and red being very, very high overlap. In other words, showing that the correlational structure and what these modules are made of is essentially the same or highly overlapping in both these labs. And this gave us confidence that what we were looking at was something that was reproducible. Well, we looked in many other data sets and we identified 5, and we're able to confirm this. We had 2 modules that were up after nerve injury, shown here pink and magenta, and 3 that were down. And we looked in 16 independent neuronal injury data sets. And here is taking our PNS module. This is one of the modules, that magenta module that's upregulated in PNS versus PNS. And you can see a lot of red, not complete, but these are different data sets, different paradigms. This is what PNS versus CNS looks like. Again, almost no significant overlap. It looks almost random. So what we're seeing is, again, in other data sets, what we saw in our initial analysis, that there's a preserved co-expression pattern of a network of genes that forms modules corresponding to biological processes that are up and down regulated in the PNS, but not the CNS. So then we were able to reduce this to its known protein-protein interaction network. What we essentially do is we take the binary matrix of known PPIs in human. Remember, all of these studies are done in mouse, and we multiply that by the correlation matrix or the topologic overlap matrix from our other, from the co-expression. And we end up with a reduced but very, very tight network that's validated by human protein-protein interactions. The blue genes in the middle being transcription factors. So most of the transcription factors are sitting in the middle. And what happens is, if we remove the transcription factors, one can see that if we remove the core set of transcription factors here and remove most of them, the network dissolves. So what it's saying is that there's a transcriptional response. It's not one transcription factor, it's not 2, it's not 3. It's a core set of co-regulated transcriptional factors that's driving this response. There's a kind of injury response that can occur in the PNS, not in the CNS. Very, very similar to the idea that there's a transcriptional, that one can reprogram a somatic cell to be an induced pluripotent cell in some ways. So when we actually take those transcription factors and divide into kind of known pathways, we can see that what's happened here isn't one thing. There's a coordinated response of multiple biological pathways, from TGF-beta signaling, cytokine receptor interaction, JAK-STAT, chemokine signaling, neurotrophin. Many of these were known individually to be involved in single genes, but what we're showing is that this is a coordinated regulation that's necessary. At least that's the hypothesis. So what do we do with this? Can we target a network? I mean, it would be, we tried a number of ways using baculovirus to overexpress 5 or 6 of these, and it's not really technically feasible yet. So we were asking, can we target a network? How could we do that? So we used this connectivity map that at the time had 7,000 expression profiles. What they basically do is they take cancer cells and they give 1,300 FDA approved compounds. You have a gene expression pattern. So we asked, can we map any of these compounds onto the changes that are occurring here, and identify drugs that would reverse the pattern, and then you test the drugs in model systems. And so this is what actually happened. We found this drug Ambroxol, and what's shown here is what happens to the genes after sciatic nerve lesion, the red being up, and those that are blue are upregulated after this drug in cancer cells, not even in neurons. And so we said, okay, this is not recapitulating the entire network, but it's recapitulating some of the major transcription factors, quite a number of them. And so we gave Ambroxol to dorsal root ganglia in a dish, and we showed, indeed, Ambroxol after 24 and 48 hours does increase neurite outgrowth. Then Zhigang He's lab at Harvard injected Ambroxol into the eye of mice after an optic nerve injury where there's normally no regrowth, under the paradigm shown here. And he had also shown a few years before that knockdown of PTEN and SOX3, but PTEN being the most, so far, the strongest effect on neural regeneration seen. If you knock out PTEN, you get very, very strong regeneration. And so we did this on top of a PTEN knockdown. So on the left is shown the PTEN knockout, and there you can see some regrowth. This is the PTEN plus ambroxol. So again, this drug that's picked out because it is predicted to change a bunch of transcriptional programs related to neural regeneration does indeed increase that. And we've gone on to do this in a more refined way with more refined data sets and identify a number of other drugs that are acting similarly. And again, this is just quantitative comparison of those data over a bunch of different distances from the crush site, really showing substantial augmentation of growth. So again, here we identify a transcriptional program observed after PNS, but not CNS injury in rodents. The program links known signaling pathways via a core set of transcription factors that's conserved at the PPI level in humans. We experimentally and bioinformatically validate several network predictions, and we use that profile to identify a drug that promotes regeneration. So now how about problem 3? I'm going to show you another example where we do something very similarly, but in slightly different disorders. So here we used gene expression profiling in human and mice to identify evolutionary conserved gene networks that mediate neurodegeneration. And then we tested their causal relationships. So we have a bunch of diseases we're working on, all of which involve the microtubule associated protein tau. That's not, they have massive differences in clinical presentation, and there are other proteins involved, like in Alzheimer's on the left. But deposition of tau, the microtubule, in these hyperphosphorylated, it's called a tauopathy or proteinopathy, is common to these disorders. We don't know how to prevent or cure them. We don't know how different they are. We don't know what causes them mechanistically. Really, we do know a lot about the genetics. And in fact, in frontotemporal dementia, there's a tau mutation that causes, a bunch of tau mutations cause about 10% of the familial cases of frontotemporal dementia. In PSP, there's a haplotype around the chromosome 17q inversion that is present in 96% of people with PSP. So again, tau being a major genetic risk factor, much more minor for Alzheimer's disease, we found a rare variant in tau called A152T that increases its risk about 2-fold. So we worked, so here's our approach. We took, we had this notion that a lot of the problem with neurodegenerative dementias and modeling them in mice is that it's on a recessive, inbred mouse background, people only tested in one mouse strain, et cetera, and you don't know if it's really relevant to humans. So we did detailed profiling of disease pathology-associated pathways using RNA sequencing, multiple brain regions, at early stages before there's cell loss and later. And of course, in the mouse, we can look at a cell type level, and we have tools for functional validation. The issue is, in human, of course, and then we tested them in independent published data to, again, to show that what we're looking at is not, just a vagary of our experimental design. And then we sample human disease to show that what we're finding in mice is actually relevant in human. The issue with human disease is that by the time we get the samples, the disease has actually already occurred. So we're looking at the effects of cell loss, not the kind of early changes that are occurring. And so by combining all this, we get a robust species conserved signal for discovery research. And this work was led by Vivek Swarup, Flora Hintz, and Jessica Retzak, and was published last year. So we took this transgenic overexpressing one of the most common tau mutations causing frontotemporal dementia, the P301S mutation. We made F1 crosses to get rid of most of the recessive background or the inbreeding. Both on, of course, C57 black 6 is the background, so that's going to contain it. But then we outcrossed two very divergent strains, DBA and FEB. We took the F1 crosses and we dissected into 4 regions. We included the cerebellum because the cerebellum is unaffected both in human disease and in these mouse models. We ran microRNA and mRNA sequencing, and we ran WGCNA. for a total of 288 samples. So it's a huge study. And this was made possible by working with Takeda, who did all the mouse breeding and stuff for us. It was an enormous lift and quite extraordinary. So again, this looks a little complicated, but it's just giving you a sense of kind of the data that we brought to bear on this. So we have our initial experiment. We have 3 and 6 months in TPR50 master consensus network. We have mouse brains from Takeda. So here's what we started with. They had done microarrays on hippocampus. We had human frontal temporal dementia cortex from a paper from Chen Plotkin et al. We had purified P301 mouse microglia. We actually had mass spectroscopy from a handful of patients with FTD from Emory. And we had a bunch of microarray studies that we integrated into this from the literature. So we basically asked, we want to find things that are conserved across different mouse models, that are changing early before there's cell loss, and that we can see in human tissue, essentially, and we can validate at the protein level. So we ran WGCNA, and we found 2 major large modules that were conserved. One we called neurodegeneration associated inflammatory module, or turquoise module, and it involves inflammation and it involves upregulation of microglia and astrocytes. It's upregulated at 6 months, not in cerebellum, which is unaffected. We had another module that's neuronal. Of course, these are different than the ones that we saw in autism. Slightly different processes. Although, again, this shows how gene ontology is kind of really, I mean, it's useful at some level, but it doesn't have the refinement that we really need in the nervous system, just as an aside. This module here was strongly enriched with genes correlated with phospho-tau in the model. The odds ratio was 42. So it's good to have a comparison region because we could see that these changes were only occurring in the affected regions. We then looked in multiple other data sets. Human PSP cortex in which RNA sequencing had been done, human temporal cortex from Alzheimer's, Alzheimer's frontal cortex, and at the protein and transcriptional level. These were preserved and they were regulated in the same directions as in the mice. Then we looked at some other comparisons just to make sure it wasn't a postmortem artifact, looking at bipolar, schizophrenia, major depressive disorder, and pathological aging, which is amyloid deposition without tauopathy and synapse loss. So we have these conserved modules, and then we asked, are they enriched for disease associated variation? And what we could see is a separation between the primary tauopathies, FTD and PSP, shown in green and red, and Alzheimer's. What we could see is that this turquoise, which is the inflammatory, is where Alzheimer's disease, GWAS, is enriched. So it's primarily an inflammatory or glial disorder, whereas these other disorders have primarily neuronal first. So it's very interesting to think about it because in the end, all of these disorders have downregulation of synaptic genes and kind of neural inflammation. And yet the causality, so they must converge at some point to some degree, but the causality starts at different places. This is just some of the known disease genes in these different modules. And the ones that are black have been identified in GWAS or association studies in GWAS for these disorders. So now we want to know, okay, what's driving all of this? So we looked for transcription factor enrichment. We didn't find anything that was remarkable to us. But when we looked at microRNA, we found something really remarkable. We found that there were a bunch of microRNAs whose targets were enriched in the salmon module, which is again, the downregulated neuronal module, the turquoise being the upregulated inflammatory module. And this one MIR203-3P was a hub of a microRNA module enriched in the salmon. And it was the most upregulated microRNA. 25% of the NAS module genes or salmon module genes are targets of that MIR, which was a remarkable, huge enrichment. And when we looked in frontal cortex of people with FTD, we saw that it was indeed elevated, this microRNA, in disease. Looked in cerebellum, it was not changing. When we looked at quantitative proteomics, we could see that the level of its predicted targets is also downregulated, as you would expect from upregulation of this microRNA. So is it sufficient to cause neurodegeneration is the question we asked. We overexpressed it in vitro and in vivo using AAV-mediated gene overexpression by injection, and we looked at gene expression in cell death assays. And indeed, we find after 6 weeks of injection of the microRNA, it's in vivo, it is downregulated, that module, and we see apoptosis increasing. So now we wanted to ask, can we modulate this at all? So we took a tough decoy, which is a way of reducing the miR203. So we showed that, number one, when we increase miR203 by upregulating it in cortex, presymptomatically, it causes neurodegeneration. When we do the tough decoy at 3 months and then look a month to 2 months later, we can see again that overexpression induces the module genes and accelerates apoptotic pathways, and the tough decoy downregulation increases the NAS module genes and reduces apoptotic pathways. So a proof of principle that this is actually driving the things that we predicted it would. So now what do? Well, we decided to take these modules, screen through the CMAP like we did before, and identify drugs with opposing gene network changes, hoping that those could block miR203-induced neurodegeneration. Actually, in our top 30 hits, 4 of the 6 HDAC inhibitors in the entire list were identified. So this told us HDAC might be something to try. And indeed, this is showing the NAS module eigengene in cultured neurons where we overexpress either scrambled or the miR203. And what one can see is that with the DMSO control, there is a downregulation of the eigengene of that module. That is, those genes go down. But when we add SAHA, 0.5 and 1, we see a progressive dose response that actually substantially normalizes the difference between those two. And we also did this with Steve Haggerty at Harvard in iPSC-derived neurons, and we're able to show a similar thing. This is just showing, the tunnel experiments here that were done in vitro with these mouse neurons. And again, we used a second drug that had been identified in the screen called scriptade. And what we see there is if you look at the tunnel positive nuclei, not just the module eigengene now, we're actually asking, is it reducing cell death in addition to changing gene expression in the proper direction? What we see is apoptosis with DMSO. But when we, and the red is the miR203 and the black is the scrambled, we see that this becomes attenuated as we add 0.5 and then 1 micromolar SAHA. What we find with scriptade, which is interesting, is that after 1 micromolar, it starts to get a little toxic. But still, we can see that we've massively reduced the percent tunnel nuclei at 1 and 2.5 micromolar. So these are 2 proofs of principle that really show that we can use these transcriptome profiles, identify regulators, and then use those to develop a mechanistic understanding and potential treatments. So comparative network analysis permits identification of gene expression patterns and drivers associated with PNS and CNS regenerative capacity, integration of cross-species genomics data, and addressing genetic background effects in mouse models, which has not really been done much before, provides a framework for identifying therapeutic targets in neurodegenerative disease. These approaches provide a framework for identifying genetic and pharmacologic modulators of neural repair and degeneration. And we think that using these transcriptional networks for drug discovery in and with coupled with psychiatric disorders is a promising frontier. So I have a lot of people to thank. We have a lot of funders, both NIA, the Tau Consortium, the PsycEncode Project, NIMH, and Takeda were the main funders of this. Vivek Swarup was funded by a postdoc award from Larry Hillblum Foundation. The neuropsychiatric disease work was led by Neil Parikshak, Mike Gendel, and Vivek. The Tau Dementia Networks, again, Vivek and Jessica Retzak and Tim Chang and Flora Hintz in the lab. Again, our collaborators, Alan Levy at Emory, who we were working on the proteomics with, as well as our essential collaborators at Takeda, who were absolutely wonderful to work with. The neural repair work, again, was done across multiple labs over almost 6 years and involved Giovanni Coppola and Steve Horvath at UCLA, Clifford Wolf at Harvard, Mark Dzinski at UCSD, Mike Fanzelber at Weizmann Institute, Larry Benowitz, and Jigang He at Harvard. Thanks for your attention.
Well, thank you very much for this very interesting presentation. We have a lot of questions, so maybe we can read them really fast. I'm sure the answer will not be so fast. So,
yeah, read them to me, please.
Okay, so Diego Arturo is asking one question. A lot of these neuropsychiatric diseases, I guess, are more in a cognitive level of the brain. Are there any hypotheses or possible etiology for this condition that can be concluded from these enriched modules?
Well, it's cognitive and behavioral, and we see a change in the state when we actually work. We can begin to ask, what biology do these modules actually represent? We don't really fundamentally know, but some of them are quite related to neuronal firing and neuronal energetics and firing rate. And so we think there's been a change in local circuits and long-range connectivity that's altered neuronal firing rate. And it might be actually quite subtle. And we need to be able to recapitulate that in animal models to really understand a little bit, or in cell culture, to understand really what the mechanism is. But one of the things that we find is that some of these changes are enriched in the superficial layer of the cerebral cortex. Those are the layers that connect the hemispheres to each other and connect regions within the hemispheres. So that fits with imaging data that shows long-range disconnection between front and back of the brain in these disorders. And the cells that project that way have the most changes. So that's pretty interesting. So there's some convergence, but we're a long way from being able to answer that question in a way that I'm satisfied with.
Okay, thank you. So I'll read the next question. The next question is it possible to decrease the symptoms in a neuropathology by attacking a hub gene? Or
well, that's what we want. Exactly. That's what we really want to know. But rather than going for behavior or symptoms, I'd first like to have something where I can screen in a dish using microscopy or some kind of biological assay, where I can say, just like we did with neurodegeneration, we could look at apoptosis, we could look at cell survival, and in neural regeneration, we could look at neurite outgrowth. So I'd like to be able to look at something, whether it's circuit dynamics, network activity and connectivity, synaptic function, firing rate. I'd like to have something like that before I would go on to measuring behavior. But it may be that we just have to go bite the bullet and do that.
Okay, so I think you are answering actually the second part of this question, that was, or is the system too complicated to show any kind of improvement without targeting a dozen or even 1,000 of genes at the same time?
Well, yeah, I mean, the interesting thing is you usually aren't targeting. I do believe we'll have to target, we'll find drugs that for some mechanism push the entire module in the right direction. And that might be by targeting a few hubs, or it might be through some other trans mechanism. Don't know yet.
So the next question is from Hiromi. So are there any specific transcriptomic patterns which correlates with the levels of severity of a neuropsychiatric disorder?
Well, it's a really interesting question. And the question is, at some level, if I reframe that question a little bit, if we look at autism, schizophrenia, bipolar, autism being the earliest onset, you could call it the most severe because it's earliest onset, schizophrenia being next, and bipolar being third, the level of changes in neuronal function, those 4 modules, and now is always most in autism, for the most part. And so there are a bunch of modules that are worse in autism, slightly less in schizophrenia, and either unobserved or much milder in bipolar. So if we're looking across the disorders, we can see that. We don't have enough clinical information on these patients to know how it's related to severity individually, but they might be.
Oh, I lost you.
Can you hear me?
Yeah, well, now I can hear you.
Yeah, I said, but they might be related. Yeah.
Yeah, I had actually very much the same question. The next one is from Itzel. It says, I know that autism and the majority of mental disorders are considered a spectrum disease. If we focus on autism, it has different degrees of severity, going from mild to severe. So when evaluating this factor genotypically, how do you take the spectrum factor into consideration when you take autism as a variable?
Great question. Yeah, that's a great question. I'm sorry, is it?
There's a second part to it. Does it genotypically present the same changes, or how do you normalize that?
Yes, so remember here, we're looking at autism that has multiple causes. And in fact, what I didn't tell you about is that we have 8 cases with a single genetic mutation called 15q duplication. And those cases share the same pattern of dysfunction that we see in idiopathic autism. So we can assume, because we know that no single gene disorder really accounts for more than 1% of autism, that when we're looking at 50 different cases of autism, that most of them have different genetic backgrounds. So remember, we're looking at the brain, we're looking at different genetics, and we're asking, is there a pathology? Just like in Alzheimer's or any of these other disorders, there are many different, genetic, there's a, differential, it's very genetically heterogeneous. And so that's what we're kind of defining. Now, the other question you're asking, though, is really fascinating. If we took different forms of genetic mutations, those that are common genetic variation that have small effect sizes, those that are inherited, those that are de novo, like Down syndrome, very large effect, de novo mutations, do those impact different pathways during development or function? The answer to that is yes, and I didn't touch on that today, but I can refer you to a series of papers where we've actually looked at that, and so have others, starting with a paper in Cell in 2013, Parikshak et al., and then there's a review in Nature Reviews Genetics in 2016, also Parikshak, Gendel, and Geschwind, and in those, we take that tack. So what we have is mutations and different forms of genetic variation are acting during development, early development, on different pathways, but there is some convergence. And then somehow, through things that we don't understand yet, we end up with a shared molecular pathology in about 2/3 of patients after they're born and live a little bit with autism. So that's the kind of challenge to begin to kind of connect those two observations mechanistically.
So I'm going to move to the next one. It says, what proportion of these SNPs occur in non-coding regions that can participate in genomic topology has been discriminated? Perhaps elucidating this could explain the alteration of the transcription of a large number of genetic elements, whether coding or non-coding.
Yeah,
that was a question from Sebastian.
Yeah, it's a great question again. So when we actually look, so we can look at this. We can ask if we have fetal brain and adult brain EQTL, things that we know regulate expression, or splicing QTL. Do those overlap with non-coding genetic risk, either in autism, schizophrenia, bipolar, et cetera? And it really doesn't explain that much. And of the gene expression changes that we see, in other words, very few of these are actually EQTL. Very few of the genetic risk, very few of the expression changes that are occurring can be related to an EQTL that's also a risk variant. There are a few, And in the science paper, we discuss a few of them, but it's a minority. So that these are secondary to changes in underlying biology, most of the transcription changes. We also, though, ran again, I didn't, for time's sake, didn't get into it. We also ran a transcription-wide association study where we actually use brain transcriptome and we impute transcription into the, into the GWAS, and we're able to identify a lot of new candidate genes. And again, some of these, a few of them, overlap with those that are dysregulated, let's say in schizophrenia brain, but it's not the majority. So that the causal variation is acting, we believe we can show in fetal brain development, and yet later leads to these changes. And again, that's that intermediate part that's hazy that we need to.
Exactly. So the next question is, what algorithm do you use for module identification?
Yeah, we use, something called a dynamic tree cutting algorithm, which is kind of relatively unbiased. I want to make a point that, a module is something you define, you come up with some cutoff. What we usually do is we define the modules because it, on the periphery of the module, those genes don't change what the module is doing at all. It's really the core genes mostly. But what we do in all of our work is we create a table where we have a gene by eigengene relationship to all modules. So you can see, because some genes are kind of on the periphery, but involved in one module and at a center of another, right? They're not, not fully distinct. So we actually, although we simplify by kind of cutting around these modules, the entire network is more complicated than that. And we kind of provide tables that allow one to actually look at that. So it's a very good point. But we use dynamic tree cutting, which is an algorithm created by Peter Langfelter and Steve Horvath, in which they've shown is relatively kind of an unbiased way to get the most robust modules. There are a lot of different ways to do that.
Okay, so let's move to the next one. It's a question from Diego Arturo. He says, regarding regeneration, is there any interaction with glutamate? Because I know that there is an increase of extracellular glutamate in CS that triggers glial scarring instead of regeneration.
You're getting into that's a great question. It's a very, very complex issue. Some of my collaborators in this network that I described, including Michael Safroniev, have done a lot with the glial scar. In fact, there was a recent paper in Nature last year that I urge you to go read, which really gets a lot to the mechanism of what this glial scar is really doing. It's pretty unclear to me, and I don't think the evidence is that strong anymore, that the glial scar per se is actually blocking regeneration. And that work kind of shows that. There are two things, if I, just to simplify this, and I can only, you can think of regeneration as two things. One is the intrinsic state of the neuron, which is what we've been studying. And the other is the extrinsic factors that may be blocking. You may need bridges and other things like that. And so we're looking at the intrinsic state, not ignoring the extrinsic state, but with, again, this notion that it's a kind of reprogramming problem that, very similar to iPSC reprogramming, that you can change the transcriptional state of a CNS neuron to make it pro-regenerative. And then in concert with that, you're almost certainly going to have to do some things extrinsically to make that environment work. But there's a lot of evidence in the field that really the key barrier is really intrinsic. So glutamate, glutamate doesn't, again, it's complicated, but I'm just going to tell you to stay tuned. Within the next year, we'll have a paper coming out where we do show that a neurotransmitter has an enormous effect on regeneration, but it's not glutamate.
Okay.
So that's all I can say.
All right.
Top secret. Yeah.
That's what it looks like, but we will keep posted. Okay, the next one is from Erika Montserrat, and she asks, there are currently co-expression networks prediction methods that take into account the isoforms such as SpliceNet. Would it be worth running this method to have a more refined analysis?
Yeah, so we actually do that. I didn't go through the splicing networks. One of the points I want to make that I didn't make, and thank you for bringing this up, is that what we saw in the large psych-encode transcriptional study, the Gandel et al. Study, published in 2018, was kind of, in a way, I don't want to say surprising, but it was pretty large effect that when we looked at splicing effects or isoform effects, which include splicing and alternative start sites, right, alternative transcripts, we could, alternative promoter usage. So there's alternative promoter usage, there's splicing, and those things come together to form isoforms. So we looked at isoform and splicing. We found that had a stronger disease-specific effect. In other words, when we looked at schizophrenia versus autism or autism versus bipolar, isoforms distinguished them more than just gene expression. And when we looked at the cell type specificity of the modules, we saw the same thing. So I think that isoform and splicing is where a lot of the meat is. And I also refer you to a paper that a former grad student, Rebecca Walker, published in 2019 in Cell on fetal brain EQTL and splice QTLs. And there we show that splice QTL actually harbor as much, if not more, of the genetic risk than EQTL. So I think your sense that isoform is important and splicing is important is right on. It's extremely important, and it challenges us because it means that we're going to have to understand what these different isoforms actually do at the protein level, which is quite difficult.
So the next question is, have you analyzed neurogenesis within neuropsychiatric disorders? If so, how does the general outlook change when comparing with a normal condition?
Yes, and so in several of the studies we've done where we ask, where does genetic variation, both in schizophrenia and autism, and even in bipolar and depression, when we look at depression, ADHD, and schizophrenia, we can see that common genetic risk is enriched in enhancers that are open, mostly inactive, at the peak of neurogenesis in the fetal brain. There's a huge enrichment there. When we look at the genes that are mutated by de novo mutations, we see the same thing. When we looked at this EQTLs in fetal brain, we could see that there's a module that's very, very enriched in autism risk. Again, it's expressed during early fetal development. So a lot is pointing to neurogenesis of glutamatergic neurons during fetal development as one key area that gives risk for psychiatric diseases. In fact, in a 2016 paper, Hei-Jung Won, who was a postdoc in the lab and who now is at UNC Chapel Hill in genetics and neuroscience, she did the first kind of high C study, chromatin structure study of a human tissue. And we did this of fetal brain. And one thing we were able to show is that by using those data, we were able to re-annotate the schizophrenia GWAS. And one pathway that was discovered was neurogenesis. So again, multiple lines, even in adult disorders, leading to neurogenesis as one component of these disorders. So yes, great question. And you're right on. I don't know how you knew that, but great. Neurogenesis is really important.
Okay, so there is a more general question from Sergio Encarnacion. He asks a general question: what is the percentage of correlation between proteomic and transcriptomic modules?
Yes. Well, one of the problems with answering that well is that often we're comparing different studies done in different laboratories with different tissue. So like I take fetal brain, I might run transcriptome, somebody else might run the proteome. So it can be a little hard. In a recent study in Cell Reports, it's Swarup et al., we actually had the ability to agglomerate all of the Alzheimer's proteomics data that had been produced in the US with transcriptome data from the Mayo Clinic site. So it's about 70 cases or so. And we're able to see, if I remember correctly, it's somewhere around a 0.6 R squared. And between the proteomics co-expression and transcriptome co-expression. So it's around 0.6 or 0.7 is generally what people say. And so in the majority of cases, it's going to be correlated. But if you're if you're studying a single gene, it may not be. And so therefore, if you're interested in the protein, you have to measure it.
Okay, so the next question, we only have 2 more questions to go. I have a question. I was reading about, that's a question from Eli Sulvaran. I was reading about Krauss' hypothesis, and I'm very curious about the fact that there could exist a correlation between human brain evolution regarding to linguistic processes plus and schizophrenia. Therefore, that human lineage-specific genetic variation giving rise to advanced cognitive skills could be correlated as well. How strong is the human-specific genetic variation load to not only schizophrenia, but other well-known psychiatric disorders?
Yeah, it's a very interesting question. So I'm going to punt that a little bit, but I'm going to refer you, and I'm not trying to self-promote, but I'd love to get your feedback on this directly. I gave a talk, the plenary lecture at the Allen Frontiers meeting, maybe like 2 or 3 years ago. So if you look up YouTube, Allen Frontiers, my name, I gave a talk exactly about this, about autism. It's not really about schizophrenia per se. It's about genetic, how to conceive of disorders, right? We didn't evolve to have schizophrenia or autism, right? So they're obviously, and the question is why they persist in the population, which is what Crow is driving it. And so I kind of deal with that using modern genetic findings and looking at variation in IQ tests and cognitive function and what's been published. And so I'd love you to go take a look at that. And I'd love to have a dialogue with you on that because I'm really trying to wrap my head around it as well. So if you're really interested in that, I actually do have a kind of talk where I've focused on that issue because I think it's extremely interesting. Like, why do we have these in the, after hundreds and thousands of years? So, notions of balancing, selection, et cetera. I can say that there have been, when you look at the genetic correlation of human traits, there's a nice, huge study in Science led by Verneri Antilla from Broad, maybe 2 years ago now, called, what's it called, Brain Something. And it's a big consortium where they basically look at the genetic correlation across conditions. And autism is positively correlated with educational attainment, schizophrenia negatively. That's pretty interesting because it would, but another interesting thing is that educational attainment is inversely correlated with fecundity, that is the number of offspring. So anyway, so there are all kinds of interesting issues out there. I urge you to look at that paper, which is the most comprehensive look at human brain traits and their genetic correlations. I don't see much correlation between schizophrenia and reading ability, language traits, positive. It's mostly negative, educational attainment.
So, Eli, you have some more work to do.
Yup.
Hiromi has one more question. I read that there has been shown a correlation between the gut microbiome and neurological disorders. And I was wondering what was your opinion in this novel researching line as a neurobiologist?
Wow. Yeah, that's a loaded question. You know, I would, yeah, I guess I would say the following. I was incredibly skeptical about this, especially given how the studies were done. You know, there are a lot of issues. Many of them don't control for a lot of very important factors, like how the microbiome changes maternal behavior, and therefore maternal behavior is actually what's going on and changing the offspring, et cetera, all kinds of things like that. But there have been some very elegant studies done in the last 3 or 4 years that actually deal with those confounders. I still think there are some confounders, but I think we cannot ignore these data and we have to look at them skeptically, but I think we have to keep them in mind. You know, when we find things that don't fit into our map of the world, we need to change our map of the world, not keep the same map. And kind of, I always think of Helicobacter pylori and ulcer, right? You know, the idea that an ulcer wasn't just from stress, it was from a bacterial infection, right? And that you can treat it with antibiotics. I mean, it just didn't, it seemed wacky, right? You know, at some level, because we already knew what did it. Yeah. So so I do think that it's very, very important to, I think the microbiome is something that now we can assess very rigorously, and it's likely it's to have huge impacts on human biology. And if it's having an impacts on the brain, perhaps through gut brain, you know, the vagus nerve, you know, there's been stuff in Parkinson's that's showing Parkinson's can start in the gut and, you know, with synuclein deposition, and that can actually travel up the vagus nerve into the brain. You know, I mean, there's really just extraordinary findings, right? Things that are really novel. And I do view that area as really novel. It just has to be looked at skeptically, and one has to understand the confounders that can be occurring, especially with regard to studies about transgenerational transmission and stuff, and the role of changes in the host behavior and other things that could be transmitting rather than just the microbiome itself. But when done properly, there's beautiful work from Sarkis Mesmanian at Caltech that I think is relatively strong. It's very, very good work.
So I just have one question. Now, you have shown us very, very interesting data about all the molecular mechanisms that are underlying all these psychiatric disorders. So how much has that gone into the clinics now and into new treatments, or will it go soon, or how is that?
Yeah, so the way it's kind of working is as there's a confluence, especially let's say in schizophrenia. Now we know that there is common genetic variation and actually some mutations in some NMDA receptors in schizophrenia, or in the SYNGAP protein, or things like that. A bunch of biotechs and pharmaceuticals can drug those things, and so they're developing drugs, and there'll be clinical trials based on these genetic findings coming out and expanding over the next several years. So I think it's led to the genetic findings. Whether or not it's going to be successful is an interesting issue. But if it's anything like cancer, where the genetic findings have led to successful drugs, my sense is that these genetic findings are leading to drug development, and that will transform the treatment of these disorders, because we'll have targeted molecular therapies, not just treating symptoms broadly, but treating the underlying mechanism. And kind of like cancer, I mean, if you think about cancer, we were treating the kind of symptoms, or the big picture. We were treating proliferation and invasion. What were we doing? Antimitotic agents that stop cell proliferation that are very toxic, and radiation. And that had some efficacy. It's effective. And so most of the therapies now are kind of like that. They may not be treating, you know, they're treating the symptoms, not necessarily the underlying kind of causes. And when you get into cancer and you can treat the mutation, you get much fewer off-target effects, much less toxicity, and hopefully in a few percent of cases now, treatment of cancers that were untreatable before. And so I'm hoping the same thing happens here.
Yeah.
It's just neuroscience is always 10, 15 years behind cancer, at least.
All right. Well, but it's very encouraging, I think.
Yeah, I mean, a lot of these pharmaceutical companies that had dropped neuroscience programs have restarted them based on these genetic findings.
So that's very encouraging then. Okay, so I just would like to thank you again for being with us yesterday and today. And thank you for all your patience and all your in answering all these multiple questions. And it was very, very enriching for all of us, I think.
No, thank you. They were great questions, and thank you for the questions and for paying attention. It was great. Thank you. My pleasure.
Thank you so much. All right.
Everybody take care.
Thank you. Take care, you too. And stay home, as they say.
Yeah, well, and when things, invitation to students to come visit us when things open up. See you later. Bye-bye.
Bye-bye. Listo, Alfredo.